All right, let's jump right into this explainer. Today, we're looking at bovine mastitis, but we're not just looking at it as your standard bacterial infection. No, think of this as a costly agricultural crime scene, one that requires a seriously keen microbial detective to solve. You see, when a dairy herd gets compromised, figuring out exactly which microscopic culprit is responsible, well, that can literally be the difference between a quick, easy resolution and a massive, persistent farm-wide crisis. So we're going to walk step by step through the exact diagnostic workflow used to identify these tricky pathogens.
Here's our roadmap for today. We'll start with the Mastitis Mystery, move into gathering microbial clues, hit the catalase crossroads, spend some time differentiating the big three, and wrap up with exactly why identification matters.
Okay, part 1, the Mastitis Mystery, setting the scene at the farm.
So imagine a farmer notices the classic clinical signs in a cow. Inflammation of the mammary tissue, maybe some visible clots in the milk. We know right away there's an intramammary infection. But who's the specific microbial culprit? How exactly do you catch an invisible culprit? Well, the absolutely critical first step happens right there in the milking parlor, collecting an aseptic milk sample. Because let's face it, a dairy farm is literally teeming with background bacteria. If we aren't super careful collecting that milk, we risk contaminating our primary evidence with random environmental organisms. Plus, the target pathogens we're actually looking for are surprisingly delicate, so that sample needs really prompt, safe transport straight to the lab.
Moving to part 2, gathering microbial clues and where the forensic funnel begins. Once we're in the lab, we take that raw evidence and basically pass it through a forensic funnel. We're shrinking the pool of suspects with every single test we run. First up, we immediately culture that aseptic milk. To coax these delicate little organisms to grow, we streak the sample onto specific media, usually blood agar, Edwards medium, or McConkey agar, and incubate it aerobically at exactly 37 degrees Celsius. Fast forward 24 to 48 hours, and we're looking for these small, translucent colonies. We grab one, do a quick gram stain, and pop it right under the microscope. And boom, here is where we get our first major visual clue. Under the microscope, we are hunting for gram-positive, spherical cells arranged in chains. This is such a brilliant biological fingerprint. The reason they form these super distinct, recognizable chains is actually because cell division for this specific group of bacteria happens in just a single plane, and the daughter cells, they just remain attached to one another after they divide. So if you're looking down the lens and you see those gram-positive chains, you know for sure you're on the right track. Which brings us to part 3, the catalase crossroads, branching the investigation. All right, so we've hit a diagnostic crossroads. Seeing those gram-positive cocci in chains is awesome, but we have to make absolutely sure we aren't chasing the wrong suspect entirely. Enter the beautifully simple catalase test. We literally just apply hydrogen peroxide to our bacteria. If it vigorously bubbles, well, they produce catalase, like Staphylococcus, which, by the way, forms grape-like clusters under the microscope, not chains. But if our test is catalase negative, so no bubbles at all, and we're seeing those characteristic chains, we have successfully cornered the Streptococcus genus. Just like that, we've narrowed our suspect pool down massively.
Part 4, differentiating the big three, also known as the suspect lineup.
In the specific world of bovine streptococcal mastitis, our suspect lineup really comes down to the big three: Streptococcus agalactiae, Streptococcus dysgalactiae, and Streptococcus uberis. Now here's the catch. They all look completely identical under a microscope, so we have to rely on very specific biochemical fingerprints to tell them apart. A classic example is the famous CAMP test. We streak a line of our mystery Streptococcus perpendicular to a line of Staphylococcus aureus on a blood agar plate. Now watch this. Streptococcus agalactiae actually produces a unique factor that enhances the breakdown of red blood cells caused by the Staph aureus. So right where the two meet on the plate, they create this highly distinct, incredibly satisfying arrowhead-shaped zone of clearing. It's really cool to see.
Basically, this gives us our ultimate biochemical cheat sheet to identify the specific culprit. Think of it like a logic puzzle. If the isolate is CAMP positive with that clear arrowhead shape we just talked about, boom, case closed for the moment. You have Streptococcus agalactiae. But if it's CAMP negative, we move on and check another test, asculin hydrolysis. If it hydrolyzes asculin, you've got Streptococcus uberis. What if it's negative for both? Then we pull out Lancefield grouping. That's a serological test based on the antigenic differences in the bacterial cell wall. If our remaining suspect falls into group C, we've officially caught Streptococcus dysgalactiae.
By following this logical, step-by-step flowchart, we completely crack the case. And finally, Part 5, Why Identification Matters, and Getting to Case Closed.
So, you might be thinking, why go through all this trouble in the lab instead of just handing out some antibiotics and calling it a day? Well, because knowing the exact bug tells the farmer how to fix the farm's entire ecology. It is absolutely crucial. These 3 pathogens have totally different habitats. S. agalactiae is highly adapted to the mammary gland. It lives directly inside the milk ducts. If you find this, you're dealing with a contagious cow-to-cow transmission problem right in the milking parlor. S. dysgalactiae, on the other hand, lives on the mammary skin and in the immediate environment. And S. uberis? That's a pure environmental opportunist. If a herd is suddenly plagued by S. uberis, the farmer doesn't really have a contagious cow problem. They have an environmental hygiene problem. They need to completely overhaul their bedding, mud, and manure management. See? The lab test literally dictates the entire farm management strategy.
From an inflamed udder to finding chains under a microscope, all the way to an arrowhead of clear agar, this workflow is just a perfect example of science directly solving real-world messy problems. It's fascinating, right? So next time a dairy herd falls ill, will you know where to look? Because now you've got the exact workflow required to trace those microscopic clues and catch the culprit.